now have two presentations by young researchers. The first one is the first person to come up and give a talk is Dr. Shivaranjani Moharir from TIGS, from the Tata Institute of Genomics and Society. Shivaranjani, please come over.
Yeah, so I think this is, okay. So I am, good morning, I am Shivaranjani Moharir from Tata Institute for Genetics and Society, and I would be giving a flavor of how environmental surveillance can be used to monitor pathogens and how and pathogen associated traits like antimicrobial resistance. So my talk would be broadly divided into three parts. The first two would be the source of sample would be wastewater, and the pathogen that I would be talking about would be SARS-CoV, and then how pathogen associated trait, that is AMR or antimicrobial resistance, can be or signatures of AMR can be monitored through wastewater based epidemiology. And in the third section, I would briefly touch upon another type of environmental surveillance, which is air surveillance. I won't show the data for it. So, though wastewater surveillance had been widely used for polio surveillance, even NCDC uses it, but it came into limelight during COVID time. So, back early during when there were only a few cases that were detected for SARS-CoV, a Chinese group for the first time detected SARS-CoV in different kinds of clinical samples that included nasopharyngeal swabs that we all know of, and also stool samples. So after that, another Australian group for the first time confirmed the detection of SARS-CoV in untreated water in Australia. So this study basically acted as a proof of concept for establishing wastewater surveillance for SARS-CoV at a community level. So now it's well accepted that both symptomatic as well as asymptomatic individuals shed the viral fragments into wastewater, and if we are able to process this waste water sample, basically concentrate it, extract nucleic acids, do RT-PCR with the sample, one can actually estimate SARS-CoV RNA load in the wastewater, and based on that one can estimate the disease prevalence at a community level using some mathematical formulae. And if we are able to sequence it, one can actually track the variants that are present in the community. So it's not just the variants that show symptoms, but also those variants that do not show any symptoms. So I'll briefly talk about the strategy here. So what we are doing is we are monitoring 18 open drain sites in the city, in a metropolitan city, and these sites are sampled every week, once every week for SARS-CoV. The samples are sequenced to identify the SARS-CoV variants, and the data is used for mathematical modeling. And since I don't know why this is not. Okay, so since 2021 we have monitored these sites for around 125 weeks, and around 2800 samples are analyzed. And in the next part of the talk, I would be talking for AMR. So the same sites are sampled once a month to look at the antimicrobial resistance signatures in those samples. So basically what we look for are the genes, or because this is genomics data, so antimicrobial resistance genes, the pathogens that carry those genes, the resistant drug classes, and the mechanism that is used by these pathogens for getting resistant. So this slide basically shows the SARS-CoV RNA load across the sampling locations, and we can see there are 4 different peaks in the sampling window, 2 of which correlated with the clinical case load in the city, whereas the last 2 didn't, because by that time the clinical testing had stopped. So this is just to give an idea that even the kind of data that wastewater surveillance or environmental surveillance can give, that even if the infected individual doesn't go for testing, the pathogen that causes it can be, the prevalence of that pathogen can be estimated. So Dr. Sanjay Lamba from TIGS has used this data for mathematical modeling, and he would be presenting it on poster number 50. So I'll not talk about that data. So like I mentioned, if we are able to sequence the variants from this sample, one can actually identify the variants. So this first panel is of clinical samples, and the lower one is of wastewater samples. One can clearly see the transition of delta, that is orange or yellow, to Omicron, which is purple here. Also, if you look at these variants, the number of variants are more, or the sublineages are more in wastewater samples as compared to clinical samples, again suggesting that not all the sublineages or variants can show symptoms in humans. That's why we are able to see those variants in wastewater which are contributed by asymptomatic individuals, and only those that go for clinical testing are picked up in clinical surveillance. So recently, or back in November 2023 or December, we could also detect the traces of JN1 variant as well in wastewater. Now from here, I'll shift gears to detecting the pathogen associated traits, which is antimicrobial resistance. So before that, why study AMR? So AMR is a global concern that affects humans as well as animal health. So we have a general notion that it's the health sector or human health sector that contributes to AMR. But along with the health sector, there are other sectors or other fields as well, like agricultural practices, aquaculture, food industry, that result in wide dissemination of antimicrobial resistance across diverse habitats. So as per WHO, AMR is declared as top 10 public health threats to humanity. And a study published in 2022 by antimicrobial resistance collaborators estimated that in the year 2019 worldwide there were 4.95 million deaths that were linked to bacterial AMR, of which 1.27 were specifically attributed to bacterial AMR. And from this number, around 0.93 million deaths were caused by 6 leading pathogens that are known as escape pathogens, which are E. coli, Staphylococcus aureus, Klebsiella pneumoniae, Streptococcus pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa. So, if appropriate measures are not taken, a study reported that by 2050, annual death toll of 10 million would be attributed to AMR. So also AMR is not just affecting the health, it's also affecting other sectors. So according to World Bank report, AMR could lead to 7.5% decline in livestock by 2050, and it could also, and the world could lose around 3.8% of its annual GDP by 2050 if appropriate measures are not taken. So thus it is imperative that appropriate measures should be taken to understand this problem, and immediate attention and actions are taken towards geographical surveillance of AMR to identify the classes of antibiotics that are on the verge of being innocuous. So an attempt towards this, what we did was we collected wastewater samples from 17 sites in a metropolitan city. So these are basically open drain samples which receive influx from different kinds of sectors, including human communities, industries, livestock, agriculture, animals. So the samples are concentrated, nucleic acids are extracted, and those nucleic acids are used for shotgun sequencing on Illumina NovaSeq 6000 platform. After QC, the data is analyzed to identify the antimicrobial resistance signatures. So we have 4 parameters that we look for. The first is the genes, because this is genomics data, then the pathogens that are carrying those genes, then the drug class to which the pathogen has got resistant, and the mechanism by which resistance is gained. So this slide broadly shows the landscape of the city across these sampling locations. In the subsequent slides, I'll show it in more detail. So yeah, so we asked certain specific questions out of the data because the data is very huge. So the first question was which antimicrobial resistance genes are seen in the city across the 17 locations. So again, this plot, so this shows the normalized number of reads per million for ARGs against the sampling locations, and we could see that the 23S rRNA with mutation conferring resistance to macrolide antibiotics is the most prevalent ARG that is seen in the samples, followed by 16S rRNA gene conferring resistance to aminoglycoside, then again 16S conferring resistance to tetracycline, followed by 23S rRNA conferring resistance to lincosamide, and then the 5th one is there. So of interest, the top 4 belong to the translational machinery gene, that is the rRNA genes. So this is something of interest that can be looked in future. Why is it so? So the next question is, which drug class has gained maximum resistance in the city across the 17 locations? And based on the data, we could see that macrolides have gained maximum resistance, followed by aminoglycoside, then tetracycline, lincosamide, and finally alphamycin. So now, which pathogens are carrying these ARGs was the next question, and how is their distribution across the different sites? So Unlike ARGs and drug classes, pathogens do not show any specific pattern, which is expected because pathogens are, the prevalence of pathogen is expected to be more correlated with the kind of the location it is, the kind of influx it gets. But broadly one can see that H. pylori is marginally more prevalent over others, followed by Campylobacter jejuni and then E. coli and Neisseria gonorrhoeae and Neisseria meningitidis. So the next question of course was which resistance mechanism is most common, and it appears that antibiotic target alteration is the major resistance mechanism, followed by antibiotic inactivation, and these are the top 5 ones. So now, so this was with respect to one city in the country. How is the scenario with respect to the global scenario? There was a study published in 2019 in Nature Communications wherein 79 countries were monitored for the AMR landscape, and the data shows that macrolides have developed maximum resistance across global, across all the countries, and the same group analyzed 101 countries, and this data is a temporal data for 3 years, and again here also macrolides appear to be more, have developed more resistance as compared to others. So now we looked for, so WHO and ICMR has a critical pathogen list with respect to AMR. I am not going to take the names of this because it would take long. So basically we looked for these pathogens. In Indian pathogen list, in the high priority one, along with this there is Neisseria meningitidis as well, which is included, and recently WHO has updated their list and have put multi-drug resistant MTB as well into the list, and then we looked at with respect to our data how it is. So what we see is all of these pathogens are seen in the samples except for Haemophilus influenzae, which is not there, and pathogens like E. coli and Pseudomonas have gained resistance for multiple drugs either individually or in combinations, and also the third generation of antibiotic resistance is also seen, the resistance to carbapenem and cephalosporin is also seen in the samples. So next question was how is the pattern across the city, and the PCA analysis shows that all the 3 parameters that we looked for clustered together except for the pathogens, which was expected. Then we did a small exercise wherein we plotted on the population density map of the city the sampling coordinates and counted the number of hospitals in the 2-kilometer radius of the sampling point, and we saw a positive correlation between the ARO load and the number of hospitals that are there. But again, hospital or healthcare setting is just one kind of sector that is contributing to AMR. The picture is much wider and broader. So we have a poster, Manas, who is sitting here, poster number 31, the data would be explained in detail there, so if somebody is interested they can go. So the next obvious question was, this was snapshot data, temporally how are the patterns? So what we see is, so we have started collecting samples, and now it's almost 2.5 years that we have samples from every month, and we are interested in looking and going till 3 years. So what appears is that broadly the drug classes that have developed resistance are same across time, as in they are not showing any seasonal pattern. But what would be of interest is to look at the individual ARGs, what kind of mutations they have gained and how they are evolving with respect to time. This would give insights for developing newer drug classes. So I will not summarize this, but briefly I'll just touch upon another aspect of environmental surveillance without showing the data, which is air surveillance. Again, this work had its starting from COVID times, wherein it was believed earlier that SARS-CoV is mainly transmitted through contact, but the number of cases that were going high could be justified only if there was airborne transmission. Also, the antecedents of SARS-CoV-2, which is SARS-CoV-1 and MERS-CoV, they were reported to be airborne. So we questioned whether SARS-CoV is airborne as well. So we had 3 strategies in which we collected samples from high-risk settings, then which included hospital, COVID wards, ICUs and other such high-risk settings. Then the second scenario was closed room, wherein we requested COVID positive individuals to spend some time in the closed rooms just to ensure that the air that is there and the pathogen contribution is by those individuals. And the third is home quarantine kind COVID individuals. So in all the 3 scenarios, we could detect the virus in the air, and the load of the virus was directly proportional to the number of individuals who were there in the room. And at the end, though not for all samples, but from few samples we could culture, we could infect vero cells in culture, suggesting that the SARS-CoV in air was infective. So based on this, we want to understand the biodiversity in air in different kinds of niches, which would include high-risk settings like hospitals, then poultry, animal sector, public places. So basically, air samples would be collected and then used for sequencing and RT-PCR and for culturing. So I'm not showing the data. With this, I'm done. Thank you very much. This is the team who is involved in the work. And if there are any questions, I'm happy to take.
Thank you very much.
Thank you.
Yeah, one after another.
Hi ma'am, awesome talk. So I had a question like, so Omicron has more transmissibility.
Yes.
So it's less pathogenic. So in water, like it will be more sustained in water as compared to Delta. So will the data be biased and has adjusted for it also?
Yes, that's true. That's why I think we see Omicron staying for longer time as well, because not many people got infected, so they were not tested, but still we could detect it in the water for longer duration.
And the other question was that 23S RNA mutation that you talked about. So has it correlation with the antibiotic that is being used? So the antibiotic that targets has been used more in that area?
Yes, yes. So macrolides, so if you know azithromycin, which is commonly popped up without even prescription, belongs to macrolide drug class, azithromycin. So it does correlate. And also we looked at a data which I am not showing, the sales of antibiotics in that city for years, and that's how that group is sold more in the city, because we couldn't get the direct data of consumption, so sale is a proxy for consumption kind of.
Hello ma'am, thank you for the talk. I have two quick questions on this. So when you are making the model for the pathogen, what constraints do you impose to make it pertinent to the wastewater media you are giving, especially in terms to pH and temperature?
So we are not actually looking at the physico-chemical factors as of now, but that is something that one should look at in future. What we are using right now is just the readout, whatever pathogen load that we see with respect to, and I think Sanjay can explain it later, you can go to his poster, but the way the data set that is used is, the data is normalized with respect to the earlier time points, and that's how he is using it. You can talk to
second one is that what kind of a model you are making for the pathogen, a stoichiometric or kinetic model?
So maybe Sanjay can tell. So prediction model is what I know for.
Okay.
Hi, good talk. I have two very simple questions. First, when you do the viral quantification, load quantification, what is your normalization factor? I'm assuming some sort of fecal coliform DNA?
Yeah. So when we started this work, there is one fecal the pathogen that is used for normalizing, which is PMMOV, pepper motile mosaic virus, which is used. But by the time we had started this was not very clearly told and was not reported. So we wanted to keep everything similar. So the control that is there in the gene that we use is a human control, RPP, which is used for RT-PCR, that is there. And also for another level of normalization that we are doing is using the earlier data points for normalizing the future points. That's
And the other thing is that in one of your graphs you were talking about rRNA genes being one of the more picked up ones.
Yes.
Is that by any chance because they are more abundant?
Right. You rightly pointed out. So there are two reasons for that. One is because they are in multiple copies in the genome. Other is most of the mutations that are associated in these genes are SNPs. So the chances of picking up multiple SNPs in those genes is again higher, and they would be counted as individual events. So that's how is the other reason.
So I know these kinds of surveillance studies are now being done in other parts of India as well.
Yes.
So you have a network that exchanges the data and you collect it together for global analysis?
Yeah. So when we started, I started it from CCMB. This work was done in collaboration with CCMB. When I started, it was in collaboration with CCMB. So during early months, so we had multiple cities that were through the CSIR network. Then later on, this project was under the Rockefeller, there is a consortium, 4-city consortium, APSI.
Now even BBMP,
Yes.
even Mumbai, in the municipality corporations, they have adopted these methodologies. So do you have a network that actually collects all this data and analyzes them?
Yeah, so when we started, it was a network of scientists, and now for Bangalore, the model is well set because BBMP is very receptive. But it's not the scenario for all the cities, I would say. But yes, the network, definitely we all interact. There's a close consortium and the data is shared.
Is there a data somewhere that people can can analyze.
Yeah, it would be there. So we have a dashboard, APSI dashboard. Some of the data is password protected, but definitely we would put it out in time. Some of the part is already published also, and it's in public domain. But slowly everything would be put out.
The reason I'm asking is that public health and this data has to interface.
Yes. Actually, in fact, that is one of the main objectives of this phase now, to how to put this data in the perspective of public health and how they can pick it up and translate it actually.
Thank you so much.
In fact, for Bangalore, the model has worked really well, but for other cities, that's the to do.
Thank you very much, Shivaranjani.
Thank you.